Some of you may realize what I just showed you may be problematic for a standard textbook view, that the amygdala is the fear center. Now, there isn't actually that much empirical contradiction here, because a lot of the work on amygdala physiology was done in rodents, and typically the measure of fear, quote unquote, is just freezing behavior. In humans, we often focus mainly on autonomic responses as well, and those don't necessarily reflect conscious fear. Just as visual discrimination doesn't necessarily reflect conscious vision, as we learn from blind sight. So this turns out to be exactly Joe Ledoux's view. Some of you may be familiar with his work, which contributed to placing the amygdala in such a central place in the studies of fear. But to him, really we should just call these studies of threat rather than fear, because they may not reflect the conscious experience of fear as we know it. He has written books about it, so I won't repeat his argument here. But just to say, I find two pieces of evidence quite convincing. The first is lesion to the amygdala, even when it's bilateral, did not completely abolish fear experience. It impaired fear conditioning. We know that is the associative learning of fear. But the patient could still be afraid, for example, when she was suffocating. The other piece of evidence is based on the direct electrical stimulation of the amygdala. I first saw this from a video on social media and was very much convinced that stimulating the amygdala can cause fear. The patient in the video reported the experience spontaneously like he was mortified. But turns out such events are rare and only happened at high stimulation intensity. This seems to be compatible with the possibility that it may only happen when the activity spread to the prefrontal cortex. But still, this may seem like a rather unconventional view. Would be good to test it a bit further. So here's the prediction I didn't make, but Joe Ledoux did. The idea is that if the amygdala is really more about threat processing rather than conscious fear, then perhaps unconscious conditioning may change its activity, but not the reported subjective experience. Now then, by conditioning, I mean associative learning. That is, for example, pairing a stimulus with electric shock or a reward to change our affective responses towards the stimulus. How do we make it unconscious? Perhaps we can use visual masking to make the stimulus invisible. But like what we discussed earlier, the problem with masking is it just makes the perceptual signal too weak. When we get a null result, we wouldn't know how to interpret it. Is it because the subjective experience of seeing is abolished, or just that we don't have a lot of visual processing going on? So instead, we did something a little bit more adventurous. My colleagues in Japan have developed this method they call DECNEF, which stands for decoded neurofeedback. Sometimes we also call it multivoxel neural reinforcement, because in a sense, it isn't quite like the typical neurofeedback. I'll explain. So the idea is we apply multivoxel decoding online, that is real-time decoding during fMRI scanning. So in every trial, the subjects were given the induction cue so as to focus on their mind. But interestingly, we never tell them what to do, what to focus on. Instead, we just read out the fMRI activity from our region of interest, decode it to see if the fine-grained spatial pattern matches what we're after. If the match is good, we show them a large circle as a feedback to indicate that they will earn some money on the order of about 10 cents or so. So this is rather unlike conventional neurofeedback, in which we typically give them an explicit strategy or instruction to focus on something specific, and then the feedback signal is just some way of assisting the mental training. Here is somewhat more like a passive reinforcement learning, so it's not clear if they need to pay effort or do anything, in fact. Rather, whenever some brain pattern spontaneously occurs and it matches our target, we will reinforce it. After several hundreds of trials, typically taking a few hours spread over a few sessions of scanning, some subjects can learn to activate the right pattern. Now, you may think by then they would have figured out the strategy. That is, let's say the target pattern is one reflecting seeing a spider. Then after getting feedback for hours, they would know that all they need to do is to imagine a spider. But actually they don't. If you make them report a strategy they use, it is often totally unrelated to the actual feedback target. Even if you give them a forced choice test, say, okay, on this experiment, large feedback was given to you when your brain activity represented a spider or a snake. Tell me which group you think you are in. As a group, there will be a chance at guessing it correctly. So there's some good evidence that we have a method of inducing specific pattern of activity and pairing it with reward, all without the subject's awareness of what's going on. All they think is there's some mental slot machine and they're earning money. Now, with this multivoxel neural reinforcement method, we can test the hypothesis mentioned earlier. That is, can unconscious conditioning change our physiological response without changing subjective experience? The idea is to take the same multivoxel data from Vincent's study. Now, if we focus on the ventrotemporal areas, then of course we can decode animal categories quite well. These are not clinical patients, but even within the general population, people tend to be afraid of some of these things. We can pick a couple of them from each subject based on their self-ratings. Let's say they were spider and snake for a subject. We randomly assign one as a target and the other as a control. We do this randomization with the computer, so it's truly double-blinded. We then get the subjects to go through the neural reinforcement procedure, so every time the target representation occurs, we give the subject a positive feedback. After a few hundred trials, the question is, what would happen? Essentially, we're trying to see if we could counter-condition out the fear this way. The results are quite striking. As you see here, this is the amygdala fMRI signal. When we present to the subjects the images to the target, that is one of the two feared animals randomly chosen as a target. Before the procedure, it was positive, reflecting an effective response, just as expected. But after the procedure, this response was gone. The effect was specific because nothing like that was observed for the control. Right next to it, we have the same pattern of responses, this time for skin conductance. Now, this may seem too good to be true, but actually we have observed something like that in a previous study too, using the same multivoxel neural reinforcement procedure. That study was conceptually actually very similar, except that there we were dealing with conditioned fear for abstract stimuli rather than naturally occurring fear. So the critical finding though is this. Somewhat surprisingly, when we actually asked people how afraid they were when looking at the images of the targets and control animals, the subjective rating didn't change after the procedure. I have to repeat, we didn't actually make this prediction. In fact, it came as a disappointment. But it turns out it does seem to fit the hypothesis made independently early on by Joe Ledoux. Perhaps to change the subjective experience of fear, we need a method targeting the conscious rather than just the non-conscious process. So the previous study did not actually involve clinically diagnosed patients, but there may be some useful implications still. My colleague Michelle Krask is an expert on exposure therapy, a treatment that works really well for phobia. Based on decades of studies of animal models of associative learning, how fear can be unlearned is one thing that we can say we have rather figured out in psychology. The problem, though, is that patients often do not like to go through this kind of therapy, in which they have to encounter the feared objects or to relive the traumatic experience. So here, our unconscious method may help. Patients wouldn't even need to know what the intervention is about. There would be no subjective unpleasant feelings of fear. It is true that in our previous study, there was no effect on subjective ratings of fear. But if we could get rid of the excessive physiological responses first, then it may be easier to deal with the conscious experiences afterwards. Patients may be more able to go through conventional therapy from there. Perhaps we have a potential to address another problem too, which is relapse. The reason is in conventional therapy, the extinguished fear might spontaneously come back. That is, during treatment, patients learn that they don't need to be afraid. But once they leave the treatment session, the brain may decide that what was learned during the treatment was only specific to that context. So as soon as they go back to their everyday life, contextual renewal of fear might happen. This makes sense based on animal models of learning, which suggest that associative learning is highly context-specific as well. So perhaps our methods may help here, because we're not dealing with normal learning in the world, where there is always a context. We are directly conditioning the representations in the brain, so we may be able to bypass that problem. At least there seems to be a hypothesis worth trying. But to really bring our method to the clinic, the challenge is, how do we decode the multivoxel pattern representations in the brain in the first place? In the previous study, we did so by presenting many repetitions of the feared animals to the subjects. This is what one does in this kind of studies. So while the neural reinforcement procedure was non-conscious, but to build the decoder, subjects had to consciously see the feared objects. This somewhat sends us back to square one. Patients may not like this procedure so much after all. Turns out there's a method to solve this problem. This clever solution called HyperLightman was invented in Jim Haxby's lab. So the idea is we can infer someone else's decoder patterns based on another person's data. This is usually tricky to do because in fMRI data, the spatial patterns allowing us to decode the representational content is pretty fine-grained, and it differs from person to person. So let's say you want to find out what is your voxel pattern for spiders. It may be rather different from mine. But the trick is, if we both see some other stuff like apples, oranges, cats, and dogs, and based on these data, we calibrate our data together into a common space. That is, we rearrange our voxel patterns spatially until our decoders are similar for these things: apples, oranges, cats, and dogs. From there, through this common space, we can then assume my voxel pattern for spider will be more or less like yours after this calibration. This way, you wouldn't have to see spiders at all, and yet we can infer your voxel pattern for spiders. Now, you may think, even if this works, it can't be that great. That's true, there is some noise in the process, but the good thing is, if you think of me this way as your surrogate, that is, I see the spider images on your behalf, and through the calibration, we estimate your spider voxel patterns based on my brain data. Then, well, you can have as many surrogates as you want. Right now, we have a database of over 100 people already who have seen Vincent stimuli involving commonly feared animals. So if you do the math, we're talking about over 1/3 trials. So with so much data, we can estimate one spider pattern pretty well. And in fact, the HyperLightman procedure was already implemented in Vincent's published study because he had already anticipated this problem of how to eventually take it to the clinic. So, as we speak, we're actually running a clinical trial using this method. The National Institute of Mental Health of the United States has generously supported this research in which we're enlisting actual clinically diagnosed patients with phobia. The trial is still ongoing, so I don't know how it will turn out. But I'm hoping, regardless of the outcome, we're doing something interesting and meaningful. Part of the reason is, if you think about it, in clinical psychology, we really rarely, if ever, have this opportunity to test something in a double-blinded manner. Unlike drugs, it is often not very clear what is the placebo condition in this kind of therapy. To really make it double-blinded, not only will we have to train the clinical psychologists to give sham therapy sessions, we have to make sure that they don't know it's sham either. And that just sounds impossible. But here, we are taking the very rationale from clinical psychology and doing it directly in the brain. Hopefully, this approach may stimulate more work in the future. So, it's time for me to wrap up. I started the talk by asking a rather indulgent question. How can we ever know if other creatures are conscious like we are? Perhaps we will never know for sure. But my contention is the NCC can be studied experimentally. The conceptual issues involved are not trivial, but with some care we can make progress. My point is, despite some challenges, the prefrontal cortex does seem to be very important for conscious experiences, from simple visual percepts to emotions like fear. With the NCC, there are many limitations still. Some creatures may have brains very different from ours, and yet others may not even have a brain. But from the NCC we can start to build theories. With a plausible theory constrained by empirical data, we can start to appreciate what are the sort of neural computations that are essential for consciousness. Perhaps the NCC involves the prefrontal cortex because we need to monitor our perceptual reality. And we do this because we have developed to have brains capable of imagination and predictive coding. With this approach, we can at least make some educated guesses about what creatures may or may not be conscious, based on the best of our scientific knowledge. If 2 out of the 3 things here look delicious, and 1 look cute and cuddly, well, perhaps our intuitions are not too far off. And the science of consciousness may not be an entirely hopeless endeavor after all. So, thank you everyone. I should also thank my lab members, my collaborators, and funding agencies too.